ये लोग यहाँ कहाँ बैठ गए? Good evening all. It is our pleasure to welcome you all to the 11th Professor R.N. Chakravarti Memorial Oration. We at Department of Experimental Medicine and Biotechnology are conducting this oration every year to commemorate our esteemed late Professor R.N. Chakravarti, whose vision was to inculcate scientific aptitude in our future generations. Now I invite Professor Anuradha Chakravarti, ma'am, Head of the Department of Experimental Medicine and Biotechnology, to officially welcome you all.
Good evening all. Professor Vivek Lal, our esteemed director and of PGMH Chandigarh, our chief guest, Professor Jayaraman Gowrishankar, director IISER Mohali, the orator of the day, Professor Rakesh Segal, Dean Academics, and other professors and emeritus professors and professors. our faculty colleague, staff and student, ladies and gentlemen. Thank you everybody for being here with us today. And I, on behalf of Experimental Medicine and Biotechnology, welcome all into this oration and 11th Professor R.N. Chakravarti Memorial Oration. A special welcome to the Professor R.N. Chakravarti's family members for sharing this memorable moment with us today. Once again, my heartiest welcome to all of you, sir, to be here. Thank you.
I now request Aditi to kindly present a bouquet to our orator, Professor J. Gowrishankar, Director, IISER Mohali. I request Jyotsna to kindly present a bouquet to our chief guest, Professor Vivek Lal, Director, PGIMR. Thank you, Jyotsna. I request Sarita to kindly present a bouquet to Professor Rakesh Sehgal, Dean Academics, PGIMR. May I now request all the dignitaries to light the lamp and start the function with the blessing of Goddess Saraswati Ma. Now I request Professor Vivek Lal to please say a few words in Professor R.N. Chakravarti's honor and about this oration. Sir, please.
Good evening, everyone. It's my privilege to be a part of this August gathering today. I don't know how many of you have had the distinction and honor of having seen Professor R.N. Chakravarti work. I did. Professor Madhu Kular, my thesis guide, I must have troubled her to no end when I was doing my thesis. So I would have troubled her no end while I was doing my thesis under her. Would remember just how different experimental medicine was then as compared to today. You call it experimental medicine. I call it high-tech medicine. I call it future medicine. You understand? Anything that comes to the bedside first starts there. First starts there. That's important. Professor Chakravarti did his undergraduate schooling from Lucknow, KGMC Lucknow, it was a colossal institute, probably still one, but nowhere compared to what it was before. And he then subsequently came to PGI in 1961 or 62 after he had done his PhD from Calcutta. For some strange reason, which is still not very clear to me as a director of this institute, all the greats of this country came to PGI when PGI was born. I have no idea why it happened, how it happened, but there is some reason. The other day, the governor of Punjab was here, His Excellency Mr. Purohit, and he was complaining कि जब PGI बना तो नागपुर खाली हो गया because the best of Nagpur also came to PGI. So the best of Calcutta came to PGI, the best of Delhi came to PGI. Professor Aikut was one of them. He's given a blank check. You fill up what you want, and he tore it off. So Professor Chakravarti belonged to that league. He belonged to that league of doyens who made PGI जिसका जलवा आज भी चल रहा है. He mentored more than 200 students. I was one of them. And I can tell you one thing. He was a ruthless taskmaster. He was very particular about basics. I remember the words which he said. In basic research, in clinical research, the small things are the big things. Don't go after big things. The small things are the big things. See the data yourself, confirm it yourself. And even if it is at variance with what is known, never mind. You have said something, believe in yourself. He was the one who, I think, started the research methodology classes in PGI. He midwifed that tradition which still goes on. Yes, he was the one who started those traditions, which still goes on. Research methodology classes are today the most sought after classes, most sought after courses in PGI. And PGI is known for its research methodology classes. He did cutting edge work on atherosclerosis at a time when it wasn't so well known, but today it is well known the world over. In fact, I do not know whether you're aware or not. The father of cardiology in India, she's a lady, Padmavati, Professor Padmavati from Delhi, knew about his research and quoted it the world over. At that time, things were very different. Atherosclerosis was just coming up as a cause of myocardial infarction and myocardial events. So that's what originality is all about. So it is his memory that we honor today by giving this oration. The reason why we give an oration in honor of a person is to acknowledge his values, acknowledge what he stood for, and acknowledge what he stands for. That's important. I hope that the scientists of today remember one very important phrase, which I always tell my residents. श्रम करो तो विद्या की प्राप्ति होती है. आप श्रम करेंगे तो विद्या की प्राप्ति होगी. अगर आप विद्या का सही उपयोग करेंगे तो ज्ञान की प्राप्ति होगी. So Gyan comes beyond Vidya. Many of these PhD students, they do come to me, they have a genuine reason. हमारे साथ ये नहीं है, हमारे साथ ये नहीं, वो नहीं है. I just tell them one thing. I am for you. You are part of me. I am part of you. I'll do what I can for you. I've never looked at anyone as being a resident, as being a PhD, as being XYZ. If he's in the campus, working in the campus, he's part of a divine soul called PGI. For me, the equation begins and ends there. Professor Gowrishankar, I'm so grateful that you thought it worthy to be here. I wish you all the best, sir. Do keep coming again. I've heard a lot about you. And I'm looking forward to your oration. Thank you.
Thank you very much, sir, for such a wonderful introduction to a great legend, Professor R.N. Chakravarti. Now I request our head of the department, Professor Anuradha Chakravarti ma'am, to kindly introduce Professor J. Gowrishankar, our orator of the day.
It's my privilege to introduce Professor Gowrishankar here for the today's oration. And Professor Gowrishankar, he is the director of IISER Mohali, and we are really proud that he has accepted our invitation for the oration today. Professor Gowrishankar, he did his bachelor's of medicine from CMC Vellore, and then he wanted to do his further continue his career into the research. So he went for his PhD into the Melbourne, Australia, and therefrom he got his degree in the PhD degree from there. And after coming back to the country, he served as a leader and also as a team leader, as a scientist. And thereafter, he joined as a director of CDFT in Hyderabad, CDFT. And after from CDFT, he once again became the director. He walked into the CCMB, and later on in 2090, he joined IISER Mohali as a director of the institute. So since Professor Gowrishankar has been using genetic and molecular approaches to understand various physiological processes in the bacteria, for his outstanding research, he has obtained several awards, including Shanti Swarup Bhatnagar Award, FICCI Award, CSIR Technology Award, Millennium Award from the Indian Science Congress, Om Prakash Bhasin Award, and also the Moselio Sector Award from American Society of Microbiology. He is also elected fellow of the 3 national academies like Indian National Science Academy, Indian Academy of Science, Indian Science Academy, and also he is also in such Senior Scientist Award also. For his illustrious work, he was conferred with the Padma Shri Award by the Government of India in 2013. Today, Professor Gowrishankar shall be talking on the topic avoiding the accumulation of toxic RNA-DNA hybrids during transcription. I am confident that his experience and knowledge will benefit everyone. So now I invite Professor Gowrishankar for his talk. Thank you, sir.
So, good afternoon, everyone. Professor Vivek Lal, Dr. Anuradha, the Dean Academics, and other faculty members. It's a great pleasure for me to be here today, and indeed a tremendous honor to be invited as a speaker for the 11th Professor R.N. Chakravarti Memorial Oration. I believe that Professor Vivek Lal has given an outstanding introduction to the work and career of a giant who has spent most of his career in this institute and has brought great laurels in the field of experimental medicine, not only by the research that he undertook, but also by the number of people that he has trained who have gone on to themselves achieve very great laurels for themselves and for the country. A little bit of an autobiography here may not be out of place. It's not very often that the audience which I address for my lectures has medical people in it. And it always gives me good memories of my own career, which began in medicine. And then when I wished to go into research, there were no role models. Who do you follow, right? Anybody who does an MBBS and is reasonably good in their subjects goes on to do a very good specialization followed by a super specialization in clinical work. And so when I wished to get into research, it was quite difficult to figure out, whether one would fit into it and whether it would be enjoyable and so on. And when I got this offer to do a PhD immediately after an MBBS at University of Melbourne in Australia, the research topic, those were days well before email, and these were letters which would come 3 weeks after they were posted. The research topic was the regulation of phenylalanine biosynthesis in Escherichia coli. And I thought, my God, is this what I want to do for my career? That who is interested in this small bacterium, and that too in aromatic amino acid biosynthesis and its regulation? So from there, one has come a long way to understand that actually we are ignorant of a subject, right? So what really regulation of phenylalanine biosynthesis is really an understanding of fundamental processes of biochemistry and physiology in a model system that illuminates similar processes in higher organisms, including humans, which are far more difficult to experimentally manipulate. And this is what will be the theme of my lecture, focused on the model bacterium E. coli, but with, I hope, insights into what might be happening in physiology and pathology of other organisms, including in humans. So I begin with this slide, which is again in honor and in memory of a great researcher, clinician, and a medical scientist, Professor R.N. Chakravarti. And as Dr. Anuradha mentioned, the title of this talk is on avoiding the accumulation of toxic RNA-DNA hybrids during transcription. So I believe that all of you would have heard of the central dogma of molecular biology, and the two fundamental steps of this central dogma are transcription, where single-stranded RNA is made from double-stranded DNA by the enzyme called RNA polymerase. So I don't know whether... Yeah, so I think you can see the cursor there. So, this is followed by the translation of the messenger RNA by the ribosome to make protein as per the... Yeah, but to use the microphone and that would be difficult. Okay, so in bacteria or the prokaryotes, the central dogma has another very characteristic feature that is not found in the eukaryotic systems, and this is called the coupling of transcription and translation. So this is a very distinctive feature of prokaryotic life, and what is shown here is an electron micrograph, a very beautiful one from more than 50 years ago, that shows DNA as a strand on which RNA polymerase. So let me see this. So on which RNA polymerase has initiated transcription from there, and there are multiple RNA polymerase molecules from which the RNA molecules are seen. So you can imagine this is not in abstract. This is visual demonstration of transcription in a bacterial cell, and the RNA is completely populated by the ribosomes that are going to make the proteins. So these are called the polyribosomes. So this feature of simultaneous transcription and translation is something that is unique to prokaryotes and is not seen in eukaryotic cells because of the presence of a nuclear membrane. So because of the nuclear membrane, RNA transcription is distinct both in space and time from translation, which occurs in the cytoplasm. So the question one can ask is there a role for transcription-translation coupling other than the fact that it occurs in bacterial cells because there is no nucleus? And what it turns out is that in bacteria, there are processes for preventing the transcription that is not simultaneously translated. And these active processes involve proteins, two of which are called Rho and NusG. And what this is to show a cartoon that when RNA polymerase is transcribing DNA to form the RNA on which the ribosomes are doing their translation, in case there is a stop codon in the RNA at which the ribosomes will drop off, but the RNA polymerase were to continue, then these proteins Rho and NusG interact with the RNA and with the RNA polymerase to terminate transcription. So what this means is that untranslated transcripts are destined to be terminated. And both these proteins, Rho and NusG, are essential for the viability of bacteria. So the question then comes, why is it that Rho and NusG exist, and why is it that their functions are essential for viability? And about 20 years ago, we came up with this hypothesis based on some research that we had done. But I'll first talk about the hypothesis and then about the evidence in support of it, that the process of transcription is inherently prone to generating RNA-DNA hybrids, which are also called R loops. And these R loops are toxic, and that is shown in this cartoon. So what we are saying is that if RNA polymerase is transcribing the RNA from a DNA template, this RNA has a tendency to go back into the DNA, and because of sequence complementarity, form an RNA-DNA hybrid and displace the other DNA strand. So this is a three-stranded structure, RNA-DNA duplex and a displaced DNA strand. And the formation of this structure, or the R loop, we are suggesting is toxic to the cell. Such R loops can be removed either by a very specific enzyme called RNase H by hydrolysis, or by enzymes called helicases, R loop helicases, by unwinding the RNA-DNA hybrid. The hypothesis further says that because R loops are toxic, evolution has selected for mechanisms to reduce R loop prevalence in cells. So in bacteria, so before we come to the mechanisms, why will R loops be toxic? Theoretically, they can be toxic for three reasons. One is this structure can prevent another RNA polymerase molecule from performing transcription. So they can block the movement of succeeding RNA polymerase molecules. The same DNA template is also the template for replication for a DNA polymerase. And when this structure forms and RNA polymerase is now arrested, the R loop can arrest or block the progress of DNA polymerase. And this is what is sometimes referred to as transcription replication conflict. And in a third way, the R loop itself can prime aberrant replication initiation. So there are mechanisms by which R loops can be toxic, and consequently, there are processes to prevent R loop formation. And in bacteria, we hypothesize that there are two such mechanisms which act sequentially to prevent the accumulation of lethal R loops during transcription. So this again is a cartoon figure. So what is shown in the panel A is the default which I spoke about earlier, that in the absence of these mechanisms, the R loop will form when RNA polymerase transcribes on the DNA template. So one mechanism to prevent R loop formation is transcription translation coupling in bacteria. So because the RNA that emerges is engaged by the ribosome, it is not available to enter into the DNA double strand to form the RNA-DNA hybrid. And the second mechanism, in case the transcription translation coupling were not to occur, as would occur after a stop codon, is the presence of these proteins Rho and NusG, which will terminate the transcription so that the RNA itself is no longer made. So the corollary of this hypothesis that if we have mutants that do not have the proteins Rho or NusG, they are defective in termination of transcription, and therefore R loops will be expected to occur at increased frequency in the cells. So one way in which we were able to show that this is so is to perform an experiment using an R loop helicase called UVSW. And the UVSW helicase has been placed under an IPTG inducible promoter. And what we see here is that a strain which is deficient for either Rho or NUSG cannot grow, that is well known. However, when one adds IPTG to induce the expression of UVSW in a Rho deletion strain, there is growth, right? So we can see this. These are serial dilutions of a culture, and the culture can grow with 0.5 millimolar IPTG or 0.1 millimolar IPTG, but fails to grow in the absence of IPTG. Similarly, NUSG deletion can also grow with IPTG added. So the inference from this experiment is that excessive R loops are actually the cause of lethality, and by preventing the accumulation of R loops in these cells, one can actually confer the rescue from lethality. We have also used a monoclonal antibody called S9.6, which is very specific for the detection of RNA-DNA hybrids. And one can use an antibiotic called bicyclomycin, which is a Rho inhibitor. It is in fact approved for use for in GI bacterial infections, but is no longer available from the pharmaceutical companies. So what this shows is that if one has added bicyclomycin to a culture and then prepares a nucleic acid preparation from it, it is very strong for reactivity to S9.6. So that means when Rho is inhibited, there are a large number of R loops in the culture or in the cells, and the fact that they are R loops can be shown because the reactivity is lost when one treats with RNase-H. The same thing happens if one takes a delta Rho strain, and which is viable because the UVSW helicase is being expressed by adding IPTG. But when one removes the IPTG, one expects R loops to occur, and that's what one sees here. When one has depleted IPTG, there is reactivity, which is again lost upon treatment with RNase-H. So what this experiment shows is that the absence of Rho function leads to an increased occurrence of RNA-DNA hybrids inside the cells. We then did a genome-wide sequencing experiment to actually look for where these R loops occur. So this is the mapping of R loops. And the principle for such mapping is to use a reagent which is sodium bisulfite. And the principle is that the sodium bisulfite acts only on cytosine residues in single-stranded DNA to convert them finally to uracil, and which can then be read as thymine bases. So so if one were to take total nucleic acid from a bacterial cell, then single-stranded DNA will occur wherever RNA-DNA hybrids have occurred. And so when we treat with bisulfite, it is only in the regions of R loop formation that the C residues will get converted to T residues that can be picked up by whole genome sequencing. And that is what we did to find that there are hotspots of bisulfite reactivity which are distributed along the circular chromosome of E. coli, which is shown here. It's a very large, reasonably large chromosome of 4 million bases. Of course, it is still 1,000th the size of the human genome. But these red and blue dots or lines represent the sites on the circular chromosome where these hotspots of bisulfite reactivity have occurred, either on the top strand, which is in red, or the bottom strand, which is in blue. And the same is shown here as 3 examples of these high-ranking bisulfite reactivity clusters. And the way to try and understand this is that the bisulfite is picking up the single-stranded DNA. So, for example, in this cluster 54, it has picked up the top strand. And in cluster 59, it has picked up the bottom strand. What that implies is that the opposite DNA strand has formed an RNA-DNA hybrid. So the RNA is represented by the red strand, and consequently we can infer the direction of movement of RNA polymerase, because RNA polymerase makes the RNA complementary to the template strand. And so we have these three examples. What it shows is that these R loops are fairly long regions which span more than one ORF and sometimes are in antisense to an ORF. So for example, here RNA polymerase has moved in this direction to form an R loop, but in doing so, it has gone antisense to the ORF called YGJD, and so on. So in this next part, what I would talk about is the role of the Rho protein in preventing R loops from what is called as antisense transcription. So antisense transcription is the transcription of the strand of DNA that does not form mRNA that can be translated. By definition, therefore, it is not translated. And because it is not translated, A, it is prone to forming RNA-DNA hybrids, according to our hypothesis, and B, it is the substrate for termination by the Rho protein, because Rho binds naked RNA, which is a nascent RNA. So in the experiment that we did, what we did was to sequence, as I will show in the next slide, we sequenced all the RNA in a cell that has no Rho in it, but it is still viable because it is expressing the R loop helicase UVSW. And what we find is that the category of that we find antisense transcripts in this experiment, but they fit into two neat categories. And so I try to explain those two categories here. So if there is Rho deficiency, there is inappropriate antisense transcript synthesis. Not all of them form R loops. Those which do not form R loops can automatically be detected in a simple experiment where Rho deficiency has been created, for example, by adding bicyclomycin. However, there is a second category which form R loops, and as I said, one consequence of R loop formation is it inhibits further transcription. So these RNAs will not be detected in a simple experiment of this kind, but they can be detected in another experiment where the R loop helicase UVSW is expressed. In that case, these R loops which are formed will go back, will be unwound and will be detected by RNA-seq. And because the inhibition is removed, more of these RNAs will be formed. So consequently, what we expect is that in a strain where Rho is deficient and the helicase is expressed, a new category 2 of RNA molecules will be identified, which will represent the R loop forming RNA molecules. And that is what is shown here. These are just examples of different antisense RNA regions that we could identify from our experiment. The top two represent what we can call as either there is no expression at all, there is no antisense expression at all. All of them are very low here. The y-axis in all these cases is in the log scale. So let's keep that in mind. Or it is constitutive. It occurs to reasonably high extent in all the categories, even in a wild type strain. But these two represent what we call category 1. They are expressed simply when Rho is inhibited. And you can see here that the red and the blue are fairly high in both when bicyclomycin is added or in the delta Rho strain. But it is this category which is the novel category. In this case, only the red is very high. So that is when Rho is defective and the helicase is expressed. So you can have very high levels of expression. Even a simple inhibition of Rho by bicyclomycin does not give that inhibition. Just to give you an example, the blue line here corresponds to level 4 and the red line here corresponds to level 10. So this is a 6-fold difference. Please understand this is 2 to the power 6, which is really a 100-fold difference that is being found. So based on this is another way of representing it. So what we see here is that these are the categories of antisense expression that can be seen in what is called as a genome browser. So this is the level of expression that is seen only in the strain which is defective for Rho and is expressing the helicase. It is not found if only the helicase is expressed. It is not found if only Rho is inhibited, and it is not found in the wild type strain. So this category represents the antisense RNAs which are forming R loops in the E. coli strains. So then we have gone on to, all this work is published, and we have then gone on to ask what will happen if we can take examples of these kinds of RNAs and artificially overexpress them. So what we have done is to take several antisense regions and express them from an IPTG inducible promoter. And this is an experiment which shows several such antisense constructs that are expressed upon IPTG addition either in a wild type strain or in an RNase-H deficient strain. And what we'll find is that the things which are encircled in red or highlighted in red, you will find that they are not affected by IPTG addition in the wild type strain. They are similar here. So these, as I said, are serial dilutions, serial tenfold dilutions of the culture as we go along. But when we come here, for example, if you see 505, clearly IPTG addition in an RNase-H deficient strain is killing the strain, killing the cell, right? Indicating that R loops are occurring here, which are not being removed by RNase-H and therefore lethal. So this, and to a less extent, 503 as well. But there are, interestingly, there is another one here, 511, which is lethal even in the wild type strain. So even when RNase-H is present, it is being killed. So where we believe that it has far more severe R loop formation than the other two which are shown in red. So this is unpublished work which is being done by one of my postdocs here. So with that now I move on to considering how does this work reflect to or impinge on eukaryotic systems. So I would begin by pointing out a review which we wrote 20 years ago in 2004. Why is transcription coupled to translation in bacteria? And it is here that we advance the hypothesis that this coupling is to prevent R loop formation. But the last sentence of that, I would just read it out here. Bacterial transcription translation coupling may be viewed as one among several co-transcriptional processes, including those for mRNA processing and export in the eukaryotes that operate in the various life forms to render the nascent transcript unavailable for formation of otherwise deleterious R loops in the genome. So what we are saying in this review is that the model that we have studied in bacteria is likely equally applicable in eukaryotic systems, with the difference that instead of the ribosome being the engager of the nascent transcript to prevent RNA-DNA hybrid formation, it is other processes which occur in the nucleus, such as mRNA processing and export, which prevent R loop formation. And and subsequent studies by other groups in eukaryotes have shown indeed that this is true. This is a review from Aguilera's group in 2015, showing that In eukaryotic cells, R loops can be removed by RNase H or by a row-like, functionally row-like protein called SEN1, but they can also be prevented by the simultaneous engagement of the nascent RNA by a large number of different proteins. There is a Tho protein that is involved in nuclear transport. These proteins are involved, UAP56. These proteins, some of them are involved in splicing, and there are others which are involved in RNA degradation. More interestingly, in the last 3 or 4 years, R loops, so these are the proteins that in the absence of co-transcriptional mRNA engagement, so mRNA cleavage, polyadenylation, and splicing are all involved in preventing R loop formation by engaging the nascent transcript so that it is not available to go back into the DNA duplex. In the last 3 or 4 years, there are papers that show that the well-known genes that are required as tumor suppressor genes actually act to prevent cancer by preventing R loop mediated genome damage. So these are papers that talk about R loop accumulation being prevented by BRCA2 and BRCA1 preventing R loop driven DNA damage repair. So I just have 2 slides to show that how, what I would say, how hot this topic has become in eukaryotic research. Just as Professor Vivek Lal said that 30 years ago or 40 years ago, the correlation between atherosclerosis and myocardial vascular damage was not fully understood. In the same way, it is now turning out that 20 years ago, the role of R loops as toxic mediators of genome damage is only now being realized. In early 2000, when we published our paper, there would perhaps be 2 papers a year on R loops. Today, there are 200 papers a month, right? So this, I would just show you that this is a review which is from this year, showing the number of proteins that are involved in the suppression and resolution of the R loop here. And if this is not convincing enough, another review paper, same year, Molecular Cell, which has increased the number of proteins that suppress and resolve R loops. So I'll not go into it, but these are all papers from the eukaryotic field. So I'll conclude by saying that we believe that all transcription, both in bacteria as well as in eukaryotes, is prone to generating toxic R loops, and that one way of preventing them, which has been selected in evolution, is co-transcriptional mRNA engagement. This co-transcriptional mRNA engagement in bacteria is in the form of transcription-translation coupling, and the backup for that is row-mediated transcription termination, which also works for antisense transcripts. And this work has been done primarily at CDFD Hyderabad, but is now being continued here in IISER Mohali by Apurata. And these are some of the people. Hari was the first to identify it, as I said, 20 years ago in his PhD work. And this has been taken forward by Aisha, Krishnalila, Anupama, subsequently by Nalini, and now by Apurata. These projects have been funded by grants from the DBT and SERB. Thank you.
Thank you, sir, for such a wonderful, inspiring, and knowledgeable talk. I'm sure we have all dived into a different world of RNA-DNA hybrids. May I now request Professor Rakesh Sehgal, Dean Academics, to kindly honor Professor Gowrishankar. We are honored and thankful to have Professor R.N. Chakravarti's family join this event. However, his son is unable to join. He's unable to share his thoughts due to lack of connection. I now request Professor Rakesh Sehgal, Dean Academics, to kindly extend a vote of thanks. Sir, please.
A very good afternoon to everybody, and we are very grateful from PGI. We are all very grateful to Professor Gowrishankar, who has joined us today for such a wonderful talk, and also Professor Lal, who had already told what is the mandate of PGI and what we would like to do. So as we were discussing this research by Professor Gowrishankar, this is a sort of an interaction between, say, medicine and basic science, which very, very few people actually do. So this is really commendable, and I was listening to the talk, and I mean, was another sort of a I mean to say that I learned a lot from here, because we all read about open reading frames and what are the, say, methylation, other DNA and other things which actually lead to cancers. But this RNA loops is one new field which is very interesting, and I think he should also take it further and with this. And I'm also grateful to Professor Anuradha Chakravarti, who has invited Professor Gowrishankar for this wonderful talk. Then I see my old colleagues who have had the time to visit today's talk also. I'm really grateful to them also. And I'm also grateful to all the other faculty who have found time to come and listen to Professor Gowrishankar, and I hope they have also gained a lot from this talk. And I'm also grateful to all the people who have actually helped in organization of this oration. And as Professor Lal had said, that I also had the opportunity to work for some time with Professor Chakravarti. That was before Dr. Ganguly took over of the department from Professor Chakravarti. And I can tell you his ideas of experimental medicine were really good, and I also learned a lot of basics from him, and later on from Professor Ganguly, how we worked on the lab, because I'm also, you could say, a lab person, and we did learn a lot from Professor Chakravarti and also all the people who were there in experimental medicine. Thank you very much, and again I thank Professor Gowrishankar for this excellent work and talk. Thank you.
Thank you, sir, for your kind words. Director, sir, we need your permission to conclude this event. Thank you, sir. We request all of you to please join for high tea in the foyer. Thank you all.